Welcome to this explainer, where we're going to completely unravel the specific physics and, honestly, the biological magic that allows us to see the thinking brain in real time. We hear about functional MRI, or fMRI, all the time, right? It maps human cognition, our emotions, our memories. But what's actually happening inside that massive, noisy machine to give us those beautiful, glowing images of the brain? Well, today, we're relying on our source material to decode exactly how we capture the invisible activity of the mind. Let's dive in. So, first off, how do you actually take a picture of a thought? It's kind of the ultimate detective story. We know that everything we think or feel is driven by neurons firing. But here is the catch. We can't observe those neurons directly with an MRI. They operate way too fast, and their tiny electrical spikes just don't register on a magnetic scanner. So if we can't see the neural activity directly, how on earth do we capture and map the complex functions of the brain? To really understand the scale of this problem, just think about the number 100 billion. If we wanted to map the brain directly at the scale of a single nerve cell, which is roughly a 25 micrometer cubic volume, we would literally need about 100 billion data points just to cover an average human brain. And to catch those super fast-firing neurons, we'd have to sample that data 10 billion times a second. That is a data rate completely impossible for modern computers. You need transfer speeds of over 20 gigabytes per second. Plus, the signal from one single tiny cell would just get completely lost in the background noise anyway. So direct observation is totally out. We need a completely different approach. That brings us to section 1, the magnetic ink, blood as a beacon. Since we absolutely cannot measure those electrical spikes directly, we have to look for an invisible proxy that the neurons leave behind. And that proxy, amazingly enough, is our own blood. This whole concept relies on a massive discovery back in 1936 by researchers Pauling and Coriell. They found that hemoglobin, the molecule that carries oxygen in our blood, actually changes its magnetic properties depending on its oxygen payload. Oxygen-rich blood, or oxyhemoglobin, is diamagnetic. It has no unpaired electrons, making it practically invisible to a magnetic field. But the moment that blood drops off its oxygen, it becomes deoxyhemoglobin. Suddenly, it has 4 unpaired electrons. This makes it highly paramagnetic, actively distorting the magnetic field around it. It essentially acts as a built-in natural contrast agent right inside our bodies. Now, this chemical toggle switch creates what neuroscientists and physicists call bold contrast, which stands for blood oxygenation level-dependent contrast. The magnetic distortion caused by that deoxygenated blood actually alters what physicists call the T2 star relaxation rate of the hydrogen protons nearby. In simple terms, the magnetic field around your red blood vessels changes depending on how much oxygen they're carrying, and this directly alters the signal intensity that the MRI scanner picks up. This right here is the foundational secret of fMRI. And as you'll see on this graph, it beautifully illustrates that concept in action. When a subject inside the scanner looks at a flashing visual display, like a checkerboard pattern, for example, we can literally graph the fluctuating MRI signal magnitude over time. You can clearly see the signal rise when the stimulus is turned on, and fall right back down to baseline when it's off. We are tracking their exact brain activity simply by tracking changes in their blood. But you might be wondering, how exactly does a thought or a visual stimulus trigger this specific magnetic change?
Well, let's get into section 2, the supply chain, or neurovascular coupling.
You see, the brain is incredibly energy hungry, and it demands a really strict supply chain to keep all those neurons firing. Let's break down how this coupling works step by step. Step 1, a stimulus occurs, and local neurons fire to process it. Step 2, because they're working so hard, these active neurons rapidly consume local oxygen for energy. Step 3, this sudden oxygen consumption triggers local blood vessels to dilate and widen out to increase blood flow. And finally, step 4, this rushed delivery provides a massive oversupply of fresh, oxygenated blood to that specific active region of the brain. And here is the absolute kicker, a seemingly paradoxical fact. Oxygen supply actually exceeds demand. You would normally think that an active brain region would have less oxygen because, well, the neurons are using it all up, right? But the brain actually overcompensates. The massive rush of fresh diamagnetic arterial blood completely flushes out the local paramagnetic deoxyhemoglobin. By clearing out all that deoxyhemoglobin, the local magnetic field distortions drop, which ultimately causes the MRI signal to brighten up in that specific active area. That bright spot? That's exactly what you're seeing on an fMRI scan. Which leads us to section 3, riding the wave, the hemodynamic response. So this biological supply chain we just talked about, it's not instantaneous. Because blood is a fluid, the signal we read follows a very precise temporal wave known as the hemodynamic response function, or HRF. When a super brief stimulus happens, let's say a quick flash of light, the MRI signal doesn't just change instantly. It actually lags by about 2 seconds before it even begins to rise. Then it hits its absolute peak between 4 to 6 seconds after the event. After the stimulus stops, the signal drops, actually dipping below its original baseline at 12 seconds, before it finally recovers fully around the 20-second mark. Understanding this exact time lag is absolutely crucial for scientists to accurately map what they see on the scanner to the specific tasks the patient is doing. Moving right along to Section 4, the high-speed camera echoplanar imaging. Okay, so the physics of blood is really only half the battle here. To capture a fluid wave moving this fast across the brain, standard MRI techniques that take seconds or even minutes just won't cut it. 50 milliseconds. By utilizing highly intense gradient magnetic fields, a technique called echoplanar imaging, or EPI, can capture an entire slice of the brain in as little as 50 milliseconds. Instead of taking the image line by line over a long, drawn-out period, EPI excites the atomic spins once and rapidly acquires all the data needed for the image in a single lightning-fast shot. As the quote says, the principal advantage to EPI is the ability to freeze motion. EPI acts as the ultimate high-speed camera for the human body. With the capability to take pictures at radiographic speeds using just a single radiofrequency pulse, it literally freezes dynamic physiological processes, like the rushing of blood oxygen we just talked about, right in their tracks. Without this massive hardware breakthrough, fMRI simply wouldn't exist. But pulling off this kind of high-speed magnetic photography requires incredibly exotic hardware. The scanner needs exceedingly fast, high-intensity gradient magnetic fields. We're talking about gradients pushing beyond 25 millitesla per meter, with rise and fall switching speeds of roughly 100 microseconds. That is insanely fast. And because turning magnets on and off that quickly induces massive electrical disturbances, these gradient coils have to be heavily shielded to prevent eddy currents. If they weren't, those currents would completely destroy the EPI signal. It truly is a marvel of modern engineering. So, to wrap this all up, what we are looking at with an fMRI is just a breathtaking fusion of quantum physics, advanced engineering, and human biology. We're using massive, pulsing magnets to track the exact moments when our brain flushes microscopic areas with oxygen just to feed our thinking neurons. As we continue to refine this incredible technology, we have to ask ourselves a pretty exciting question. What secrets of the mind will these magnetic echoes reveal next? Thank you so much for joining me on this explainer. I really hope this helped you decode exactly how we see the invisible workings of the brain. Keep learning, and I'll catch you on the next one.